This is part 1 of 2 on Chapter 6, Introduction to Viruses. In the biological spectrum, viruses are considered to be acellular. So viruses are the most abundant microbes on Earth, but technically they are not considered to be living, but they do play a big role in the evolution of bacteria, archaea, and eukarya. So it's really important to understand viruses, and then in addition to their role in the evolution of living things, they also do cause diseases. So all viruses are obligate intracellular parasites. And that means that viruses are parasites, so they're going to cause disease or cause damage to a host. They're intracellular, they have to live inside of the cell of the host. And they're obligate, so that means in order for them to grow and reproduce, and to make more viruses, they have to live inside of that host cell. So properties of viruses, so like I just mentioned, they're not cellular in nature, so they're considered to be acellular or non-cellular. So they do not fulfill all of the characteristics of life. They do fulfill some of the characteristics. And in order for viruses to multiply or reproduce, they have to hijack a host cell. They have to take it over to make more viruses. So viruses are very, very tiny. So just to give you some idea, so if you can see, you have a really large blue circle in the background of this image. That is the size of a yeast cell. We also have a streptococcus cell, which is about 1 micrometer across, and E. coli, which is about 2 micrometers long. That's kind of the yellowish-orange cell on the right. So if you look, the structures labeled 1 through 10, those are different viruses. So number 2, we have the herpes simplex that causes virus. The third virus is rabies, 4 is HIV, 5 is influenza, and so on. So you can see how very tiny these different viruses are in relation to a yeast cell, which is a eukaryotic cell, and the bacteria cells, which are prokaryotic cells. So viral structure, a virus does not look anything like a cell, so it bears no resemblance to prokaryotic or eukaryotic cells. Viruses lack the protein-synthesizing machinery, so they don't have ribosomes inside of them. So here we have a little outline. So virus particle is made up of 2 parts. There's a covering and then the central core, so what's inside of the covering. And we're going to look at the covering first. We're going to look at different types of capsids, and then some viruses have a structure called an envelope as well that makes up the covering part. So in general, the capsid part, so all viruses have capsids. These capsids are made up of identical protein subunits called capsomeres. So you have these protein subunits that come together to make up this covering or this capsid. So in addition to the capsid, some viruses have another external covering called an envelope, and that's a structure outside of your capsid. These envelopes are usually stolen from the host cell, so they're not actually part of the virus or part of what makes up the virus. Those that lack an envelope are called naked, which is the virus at the top of this picture. Viruses that do have an envelope are called enveloped viruses. So these capsids, there's 2 different types of capsids. There's a helical capsid, just a continuous helix of capsomeres. It forms a cylindrical shaped virus, and you can see that shown on the picture here. And then here's more examples of these helical viruses. So they can be a naked helical virus, or you can have it in an envelope as well. The other structural type for the capsid is the icosahedral form. And this is 20-sided, and it has 12 corners, and it's still made up of those capsomeres. Besides having the capsomere that's made out of protein, some viruses can also have a viral envelope. These are mostly animal viruses, and the envelope is acquired when the virus leaves the host cell. So the virus practically steals this envelope from the host cell that was once part of the host cell membrane. And then in addition to stealing this envelope, there could be exposed proteins on the outside of the envelope, and these are called spikes. And these spikes are used for attachment of the virus to the host cell. So some viruses are going to be very specific, and then they use these spikes to attach on to the next host cell when it finds that host. The functions of the capsid envelope include protecting the nucleic acid, which is what we're going to talk about next, what's inside of the capsid. And then in addition, the capsid envelope, it helps the virus bind onto cell surfaces, and it's going to assist the penetration of the viral DNA or RNA into the suitable host. So basically the capsid envelope can help this virus particle find the appropriate host, and then it will help the virus enter into that host. Besides the type of viruses that we've looked at so far, there's another type called a complex virus, and this is an atypical virus. So one example is a Poxvirus, and this type of virus, it lacks a typical capsid, and instead they're covered by a dense layer of lipoproteins. So those are proteins and lipids combined together. Some type of viruses called bacteriophages. Bacteriophages or phages are viruses that infect bacteria cells. So these bacteriophages, they have a polyhedral nuclear capsid along with a helical tail and attachment fibers. So bacteriophages are a little more complex. And a bacteriophage, you can see a cartoon drawing of it on the right-hand side. So you have a capsid head, it's polyhedral. Inside of it you have nucleic acid. It has a helical tail, and then there's tail fibers that help that bacteria attach. So this image shown up here, this kind of summarizes what we've gone over so far. So in box A, in the upper left-hand side, we have our two complex viruses. So the first one, where instead of a capsid, it has those lipid proteins that cover your genetic material that's inside. And then the second one is a bacteriophage that I just covered. In addition, in box B, you can have envelope viruses. So you can have helical enveloped viruses shown in example 3 and 4, or you can have enveloped icosahedral viruses shown as 5 and 6 right there. Besides envelope viruses, you can have the naked viruses or the non-enveloped viruses. That is in box C, so you can have the helical capsid or the icosahedral capsid. So these are all the different types of viruses and their morphology shown up here. So if we go back to our virus particle and what makes up a virus, so far we've looked at the covering, we talked about the capsid, and then if there's an envelope, what the envelope looks like. Remember, envelopes are not found in all viruses. So inside of this covering, we're going to look at the central core next. So all viruses have to have some type of nucleic acid molecule, so some type of genetic information. In addition to that, some viruses are going to have a matrix protein, or they could possibly even have enzymes in them. And enzymes are not found in all viruses, kind of like the envelope is not found in all viruses. So the nucleic acid, in viral genomes, you can either have DNA or RNA, but you never have both types of molecules. This DNA and RNA is usually very, very small molecule compared to like our chromosomes, for example. And it just has enough genes on it to help that virus invade the host cell, redirect the cell's activity to make new viruses. So it just has enough genes to hijack that cell, or to get into the cell and hijack it. So the number of genes, it varies for each type of virus. So you can have, some viruses just have a few genes, and other viruses may have more genes. So they're a little more complex. So there's two types of viruses based on the nucleic acid. So DNA viruses, usually the DNA is in a double-stranded form, so it has the two complementary strands making up the DNA molecule. However, you can have just a single-stranded DNA virus. So double-stranded we abbreviate as DS, single-stranded would be an SS. And these nucleic acid molecules that can either be circular, like in prokaryotic cells, so you have a circular chromosome, or you can even have linear chromosomes, more like a eukaryotic cell. In RNA viruses, the RNA is usually single-stranded. However, it may be double-stranded at times. And normally we don't see double-stranded RNA anywhere except in viruses, and it's very rare. Some of these RNA viruses, it may be segmented into separate RNA pieces. So they could have like multiple pieces of RNA inside of the capsid. There's single-stranded RNA genomes. If they're immediately ready for translation, it's immediately ready to be used to make proteins. They're called positive sense RNA viruses. If that single-stranded RNA genome has to be converted into DNA first before it can be used to hijack the cell, that's a negative sense RNA molecule. So there's different types of RNA viruses within the RNA viruses. Other substances that may be found in viruses, so remember, these are other things that a virus may have, but it's not always present. So some viruses, they may have preformed enzymes that are put into the capsid along with your DNA or RNA. So examples of different enzymes, you can have polymerases. These are enzymes that can create DNA or they can create RNA. A replicase enzyme, it can copy an RNA strand. And then another type of enzyme we see a lot is called reverse transcriptase. Reverse transcriptase is an enzyme, it synthesizes a DNA molecule from an RNA molecule. And then that DNA that's synthesized, it can incorporate itself into the host cell and it can stay dormant for a little while. So reverse transcriptase, you find it in the AIDS virus. This is how the HIV can kind of hide in our cells for a little bit, so that DNA, it just kind of hides in with the rest of our DNA.